So, today we will be discussing some remaining aspects about MHC class 1 assembly and then go on to discuss MHC class 2. So, this is just to remind us that you have T cells and they are activated by MHC peptide which is present on antigen presenting cells. We did discuss this aspect about MHC restriction. What is MHC restriction? The T cell receptor recognizes MHC, a particular MHC and the peptide complex. So, the MHC is usually that of the self, and so they are restricted by self MHC, which means the peptide binds to self MHC, and this is the altered self one that is recognized by the T cell receptor. Now, this aspect is important in T cell activation, which occurs peripherally, but it is also important during thymic selection. So, for example, where do T cells learn to recognize this self MHC? This learning process is done in the thymus, and so therefore, if you do not have MHC, you do not have T cells, because the learning itself is not done. So, that is a very important aspect that students should be aware of. This for T cell selection in the thymus, you need MHC. If there is no MHC, you do not have T cells. So, for example, if there is some problem in, let us say, MHC class 1 expression, you will not get CD8s, or for that matter, if there is some problem in MHC class 2 expression, you will not get CD4s. So, it is a very important aspect that you should be sort of familiar with. Of course, in the periphery, once T cells get activated by APCs, then it will result in IL-2 and cytokine production, T cell proliferations primarily by CD4 positive cells, and this interaction also results in the differentiation of CD8 positives into CTLs, and then ultimately killing of the target cells or the ones that are presenting this MHC by the cytotoxic T lymphocytes. So, this is a broad overview of what we had discussed in the past few lectures. If you just to remind you about the structure of the MHC molecules. Now, MHC class 1, you have heavy chain, and it consists of 3 domains, alpha 1, alpha 2, alpha 3. You can see that the alpha 3 domain is interacting with beta 2 microglobulin, which is very important for the assembly of MHC class 1 molecules. So, MHC class 1 molecules need beta 2 microglobulin, and the peptide is localized, the peptide binding groove is localized in the alpha 1 and alpha 2 domains which are present over here. We had talked about some key residues that are important in binding to peptides. These are known as anchor residues, and this is typical of MHC class 1 molecules, and these anchor residues are located in this groove. Okay. Now, MHC class 1 molecules consists of MHC class 2 molecules, sorry, consists of 2 chains, the alpha and the beta, and you can see in the MHC class 2, the peptide binding groove is made up of the alpha 1 and the beta 1 domains. Okay. So, the residues that are important for binding are present in the beta 1 and in the alpha 1 domain of the MHC class 2 molecule. Just to remind you that the MHC class 1 recognizes smaller peptides, about 8 to 10 amino acid peptides. In MHC class 2, they are much longer, 18 to 20, and sometimes even larger. And as a result of which you can see the MHC class 1, what is shown over here is that the ends are closed, that the peptide is closed. So, that is why you can't have longer peptides, whereas the crystal structure of MHC class 2 has shown that the ends are open. So, that is why you have the peptides that can actually so since the ends are open, the peptide can sort of be a lot longer. Okay, just to go over the basic model of MHC class 1, you have the intracellular antigens or proteins that are generated within cells, or in case upon infection you have antigens, viral proteins for example. So, these are degraded by the proteasome, and the peptides that are generated in the cytosol need to be translocated into the ER. Why ER? Because ER is the place where the MHC class 1 molecules assemble. So, this translocation from the cytosol to the ER is done by transporter associated with antigen presentation and processing, known as TAP. So, the TAPs are localized in the ER, and these are ATP dependent, and so the peptide ends up over here. and meanwhile MHC class 1 molecules are synthesized over here. MHC class 1 plus beta 2 microglobulin, they are synthesized and they form over here what is known as the peptide loading complex, the PLC, and the peptide gets loaded on, and then the MHC molecule, there is a structural conformation changes in the MHC class 1 molecules, they are more stable, and then they traffic onto the cell surface by vesicular trafficking. Okay. So, the pathway is quite clear. So, you have endogenous proteins that are degraded by proteasomes, and then you have peptides, and then you have TAPs, and then over here these peptides bind to MHC class 1 beta 2 microglobulins and then traffic onto the cell surface. Now, I think what we should do as an exercise is to try and understand what would happen if you do not have beta 2 microglobulin or you do not have TAP. You see, now just to remind you that the MHC molecules are polygenic. Both MHC class 1, class 2 are polygenic. So, you have different forms. So, even if you lack one, maybe the other one can be expressed. So, it is difficult to get a complete loss in MHC class 1 genes, because there are so many and it is polygenic, and you are inheriting some from the father, some from the mother. But what would happen if you do not have assembly factors? I think that is an important point. So, this is what is studied. So, beta 2 microglobulin, as was shown previously, beta 2 microglobulin binds to the heavy chain, and it is important, gives it stability. So, in fact, the trimer, which is the heavy chain, the beta 2 microglobulin and the peptide, are required for proper structural integrity of the MHC class 1 molecules. So, in the beta 2 microglobulin knockout, so if you do not have beta 2 microglobulin, humans that do not express beta 2 microglobulin or mice that do not are genetically engineered such that they do not express their beta 2 microglobulin, what happens is you generate low unstable MHC class 1 molecules. Also, because you have such a drastic reduction in MHC class 1 molecules, you have very reduced CD8 positive T cells, because the CD8 positive cells are not being selected in the thymus. So, that is a very important aspect. So, similarly in TAP2, what would happen is, if you do not have the TAP knockouts, you would generate again low MHC or unstable MHC, primarily because the trimer is not properly formed. And again you would have reduced CD8 positive T cells, again for the simple reason that that again selection in the thymus is not occurring because of problem with the MHC peptide complex or lack of expression of the surface MHC peptide complexes. Now, in the previous lecture we had mentioned about a cell line known as RMAS, and RMAS was critical in our understanding of the requirements for surface MHC class 1 expression. But at that time the problem in RMAS was not discussed, and ultimately what was found out is in RMAS there is a problem in TAP2. So, the mutation results in TAP2 not being expressed, as a result of which you have unstable MHC class 1 molecules, low and unstable MHC class 1 molecules. We had also discussed another protein known as Cathepsin, or this is a protein that is associated with a TAP or TAP associated protein. And now in this case again you have low MHC class 1. Now, Cathepsin apparently has a sort of an editor function. So, it prefers loading of slow off rate peptide. That means the slow off rate means that it's allowing for high affinity peptides to be loaded on. So, it has an editor like function. Now, similarly in MHC class 2 molecules we will see a role for DM, and that is something that we can discuss. So, so Cathepsin has a editor like function. It prefers loading of slow off rate peptide. So, the slow off rate would mean that these peptides so they come off slowly off from the MHC class 1 molecules. That means they bind fairly strongly, and so the structural integrity of these peptide MHC complexes are much stronger or better. So, this was shown actually in mice that lack Cathepsin. So, we will start again and we will start off with Cathepsin. Now I had mentioned about Cathepsin. And Cathepsin is a TAP associated glycoprotein, and what it does is it helps it increases the half life of of TAPs, and it helps better loading of MHC class 1 molecules with peptides. It turns out that it has an editor function, which means it prefers loading of slow off rate peptides. So, what this means is that you have peptides that come off slowly from MHC class 1 molecules. So, in the presence of Cathepsin, what Cathepsin does is it helps MHC class 1 molecules have and the peptides have a greater structural integrity. And in fact, this editor function that Cathepsin has is sort of similar to editor function in MHC class 2 pathway, and that is primarily seen with the DM molecule. So, clearly you know it's an important aspect. Now, we had also talked about the proteasome subunits LMP2, LMP10, and in these, if single knockouts of LMP2 or LMP10 result in less CD8 positive T cell numbers, but MHC class 1 expression is not greatly affected. You just have fewer less numbers of CD8 positive cells. In the LMP7 knockout though, you have the MHC class 1 expression is low, but it is not as severely affected as in the TAP2 knockouts or in the beta 2 microglobulin knockouts. Okay. So, you have different consequences of this. It is important for students to think in terms of genes and what would happen if there are mutations in these genes, and what would be the phenotype or what would be the function. So, actual function gets shown by these methods. So, it is something that students should be very well versed with and aware about. Okay. Now, apart from the major molecules that we discussed, beta 2 microglobulin, TAP, Cathepsin, there are other molecules that play an important role, and that is primarily because the assembly of MHC class 1 molecules occurs in the ER, and in the ER there are other machinery that are also involved in this. So, some of these other ones are two proteins, calnexin and calreticulin. Now, calnexin, calreticulin are certainly not primarily associated with the MHC class 1 pathway, but they play a role in protein maturation in the ER, and as a consequence of that they are also involved in MHC class 1 assembly pathway. Calnexin is a ER membrane protein, and it was one of the first chaperones shown to bind to MHC to free heavy chain. In fact, as MHC class 1 is synthesized, free MHC class 1 heavy chain binds to calnexin. However, in cells that lack calnexin, there is no defect in MHC class 1 assembly. Now, even though calnexin binds to it, it has no obvious functional role. Now, calreticulin is similar to calnexin, except that it is luminal present, it is not membrane bound and in the ER what was shown is calreticulin lacking cells, there is some defects in MHC class 1 assembly. Of course, those defects are not as much as shown in the primarily ones that we discussed, which is beta 2 microglobulin, TAP and so on. And what was shown actually with MHC class 1 molecules are not loaded with optimal peptides. That means the peptide binding on it is not optimal. What actually happens is calreticulin is part of the peptide loading complex, and so if calreticulin is not there, perhaps the peptide loading is not that efficient, and which is why it results in this phenotype. What is important for students to understand from a cell biology perspective is that both calnexin and calreticulin are important protein folding in the ER as they bind to monoglucosylated N glycans. So, you have to think, you have to remember that MHC class 1 molecules, these are glycoproteins, and as they are being folded, if in some cases the folding is not proper, you have glucose being put on top of the terminal mannose residues, and these are recognized, and it is a sign that the folding is not proper, and both calreticulin and calnexin and calreticulin are part of molecules that bind to these misfolded proteins and help them fold properly. And that is why I have said that you should be sort of aware of these things. What happens is, while you are studying immunology, you cannot study immunology in isolation. It is a part of cell biology, physiology, all these are integrated in this molecular biology, all it's integrated, and you need to understand it, and that is why I have put this part in about calnexin, calreticulin, and the fact that they bind to monoglucosylated N glycans. Okay, so some other molecules are also important. For example, the ER57, which is a thiol oxidoreductase, and this belongs to the protein disulfide isomerase family. What it does, it helps in the proper disulfide bond rearrangements in nascent glycoproteins that are recruited by calnexin or calreticulin. So, it is part of the better folding ER folding apparatus in the protein folding apparatus in the ER that you can appreciate. Now, another aspect that I think we should discuss is the role of tap polymorphisms. Now, I had previously, when we were discussing MHC, I had said that MHC class 1 molecules are there are different aspects about MHC class 1 molecules. One is the localization, they are H2 or HLA located, they are interferon gamma inducible or cytokine induced, because then you have an amplification of the immune response. The other characteristic is they are polygenic, several genes, and polymorphic. You have several genes that are encoding that, and within the genes there are variants within these genes in different people. So, it results in polymorphisms, and these polymorphisms are useful because it helps them bind to different types of peptides. Now, what is the role of tap polymorphisms? Now, what is known is that the mouse and human MHCs are highly polymorphic, but human and mouse taps are not very polymorphic. There are some polymorphisms, but nothing at the level of which the MHC molecules are polymorphic. Now, the rat MHC or the RT1 is the rat MHC class 1 is not polymorphic, but the rat tap2 is polymorphic. And here is a very interesting observation. So, RT1A, which is this particular haplotype of the rat MHC class 1, now this together with the rat tap2A results in efficient assembly of RT1. That means they get properly loaded and you have good surface expression of RT1. Now, if RT1A is associated, or you put it in cells that contain rat tap2B, which is a different form of tap2, it results in inefficient assembly of RT1. Now, why do you think this would be? Can you just think a little bit about it? Now, it is possible that this particular rat tap2 translocates peptides that are not optimal for binding RT1A. So, you have peptides that are coming into the ER, but those peptides are suboptimal for binding RT1A, and whereas the peptides that are translocated by, or the preference shown by rat tap2A is the type of peptides is more are more the types more of the types that RT1A prefers. So, you can see as a result of which, so what happens here in this case, RT1A sits in the ER because it is not getting properly it is not getting the right type of peptides, and in the absence of the right type of peptides the folding is not proper, and since the folding is not proper, those molecules will not egress to the cell surface. Okay. So, I hope this aspect is understood. As I said, it's you know, you understand the cell biology of MHC much better if you understand the cell biology of protein trafficking, vesicular trafficking, so on. Okay. Now, the another aspect that I thought I should cover is that MHC tap proteins and viral products. Now, as I mentioned previously, viruses are present in cells, and they synthesize their polypeptides in the cytosol, so that they can make more of their numbers. Now, the immune system is such that you have a mechanism by which these proteins that are generated in the cytosol are broken down into peptides, and these peptides are presented on MHC class 1 molecules, and they are expressed on the cell surface, and this is the way for T cells to sort of peruse and get a feel for what type of what is happening inside cells. Now, you can see that one of the ways by which, and since the CD8 response is dominant, especially for virally infected cells, viruses that synthesize proteins or have recruited proteins that will inhibit MHC may be advantageous for them. It is a double edged sword, because on one hand it might save them from CD8s, but if MHC class 1 expression is lowered, it makes them sensitive to NK cells, but perhaps there is some sort of a balance in that, and from our point of view we need to understand what are some of the factors that play an important role. And over here what was found is that there are several viruses which encode proteins which inhibit the MHC class 1 assembly pathway. And as I said, over here you need to understand, if you want to block MHC class 1 surface expression, it is better to target some of the assembly factors, because then that will affect a larger number of MHC molecules rather than targeting the heavy chain by themselves alone. So, it is much smarter to target the assembly factors. So, again that is something that you should try and understand. So, I will give you some examples. So, ICP47 is actually encoded by the herpes simplex virus, and it binds to TAP on the cytosolic side and inhibits peptide binding. On the other hand, US6 is a product of the human cytomegaloviruses, and it binds TAP and inhibits TAP activity by preventing TAP hydrolysis without affecting peptide binding. You can see in these two molecules that these are two proteins encoded by two distinct viruses. They do the same job, which is inhibit TAP, but the way they do it is different. ICP47 it inhibits peptide binding, whereas over here it inhibits ATP hydrolysis without affecting peptide binding. So, you should see different viruses have evolved in different ways, and by which they are trying to outsmart the immune system, or at least the MHC class 1 pathway, by adopting various strategies. So, I think it is an interesting aspect for students to think about. So, you have some more examples. MK3 protein of the mouse herpes virus binds to TAP and it sort of targets it for degradation. The E1619 protein of adenovirus E3 disturbs interaction of MHC class 1 with the peptide loading complex, the PLC, as I said, and impairs peptide loading. So, you have different mechanisms by which these virally encoded proteins they affect MHC class 1 peptide interactions and loading, and as a result of which the surface expression of MHC class 1 will be affected. Okay. Now, one of the other differences between MHC class 1 and this is actually an important difference between MHC class 1 and MHC class 2 is that the MHC class 1 is present on almost most cells express MHC class 1. However, MHC class 2 expression is restricted, which is present on APCs, on antigen presenting cells. And these are B cells, macrophages, dendritic cells, thymic epithelial cells, so on. And these are and this bunch of cells is called the professional antigen presenting cells, and interferon gamma increases the expression of MHC class 2 in antigen in APCs and also in non APCs at sites of infection. Now, we can discuss a little bit about these antigen presenting cells, primarily because of MHC class 2, which shows a restricted cell type expression. So, macrophages, for example, they have different types in different organs, so they go by different names, and this is the name given to ones in the liver are known as Kupffer cells. These are peritoneal PECs are peritoneal exudate cells, which are present in the peritoneal cavity. You have alveolar macrophagocytes. Alveolar macrophages are present in the lung, and they ingest large particles because we take in so much dust and so on. And they produce cytokines, they help in lymphocyte activation, and they express co-stimulatory ligands, and these are aspects that we will be discussing in the T cell activation part. The other type of antigen presenting cells are B cells. Now, B cells are particular in the sense because they can endocytose the antigen with the membrane immunoglobulin that is present, and that is what makes them distinct from macrophages. In that sense, macrophages are going to be non-specific. So, they can use FCR or CD3R or the complement receptor or mannose phosphate receptors, but it is going to be non-specific. Antigen specific endocytosis is done by B cells. So, in this respect, the B cells are somewhat distinct from the other type of APCs. And so they can process this antigen once it's endocytosed, and they can activate T cells, and T cells on the other hand, they activate B cells, and so they express these molecules, and you have good interaction between T and B cells, which leads to a good adaptive immune response. Remember, the two major players in the adaptive immune response are B cells and T cells. Perhaps the most physiological antigen presenting cells are your dendritic cells, and these are present in the skin. In the skin they are known as Langerhans cells, and basically once they get the antigen, they enter into lymph nodes, and they initiate the T cell activation pathway. Now, how does one study MHC biogenesis? One critical technique that has been used in this process is something known as PulseChase experiments. I hope you are somewhat familiar with it. So, what do you mean by pulse and what do you mean by chase? So, what happens in the pulse part is that you pulse with a radioactive tracer, and for the type of experiments that we discuss, mainly cell biology experiments, you use S35 methionine or cysteine, because these amino acids would get incorporated in most proteins, and at least sulfur containing proteins, and then so that would be the pulse. And what happens is you would label them for a short pulse. So, it is always done in a short this thing 5 to 30 minutes. You wash off the excess label, the label that is not incorporated. So, you incubate the label with the cells, and so this gets taken up and it is incorporated by proteins that are newly synthesized. Okay. So, newly synthesized proteins will incorporate these labeled amino acids in it, and then you wash off the excess label. So, now these are your cells that are pulsed. What you need to do is chase, and you chase with a cold or excess methionine cysteine, so that there is no fresh incorporation of the radioactive label into your proteins, and then this can be done for different periods of time. So, you chase for different periods of time, and then you solubilize the cells with a detergent and immunoprecipitate specific proteins. So, that is basically the theory of pulse and chase, and this is a very powerful technique, and what we will do is we can discuss some experiments with you. I think it is very important, because by discussing experiments, that is how you are going to be learning you know, a little bit more about these issues. So, PulseChase is used to study biogenesis of molecules, precursor product relationships. So, for example, immature MHC class 1 molecules are found in the ER cis Golgi. And you can distinguish the immature class 1 from the mature class 1 by the sensitivity to endo-H, which is an enzyme. So, the endo-H ones are endo-H MHC molecules are immature, whereas once they mature, once they have the sialic acids and all sort of put on the MHC class 1 molecules, then they become endo-H resistant. So, as a result of which you can distinguish the immature and the mature ones. So, something very important. So, I think you can think of experiments that are around this particular thought process. In fact, I mean, these are experiments that were actually done to show the pathways involved in MHC class 1 and MHC class 2. Now, so the second example that I would come up is, you know, as I said, once you have peptide binding to MHC class 1 molecules, then they become more stable, and they can be detected with specific antibodies. So, you know, you need to think of or design an experiment how you would test the effect of, let us say, a proteasome specific inhibitor on MHC class 1 formation. Now, what are proteasome specific inhibitors? So, this inhibitor would inhibit proteasomes. Now, remember, proteasomes are the ones that generate the peptides in the cytosol. So, if you inhibit this, peptides will not be generated. If peptides are not generated, then MHC class 1 molecules, there are no peptides basically. So, it will be difficult for MHC class 1 molecules to egress to the cell surface, or even if they express, they would be unstable. So, they would be quickly internalized. So, that is some aspect that you can think about, but you know, PulseChase experiments are the type of experiments that people have done to address these issues. Now, we will start off with MHC class 2 right now. Now, MHC class 2 molecules, once they bind to peptide, you know, they are extremely stable. So, how would you demonstrate this finding? Again, we can use PulseChase experiments for this question, to address this question. So, let us say you have labeled methionine cysteine cell extract, you can immunoprecipitate your MHC class 2 molecules. Now, what you can do is, after you have immunoprecipitated, so basically you have the labeled extract, you will have beads. The beads are important because they give you a sort of a matrix, and to these beads antibodies are attached, which will recognize MHC class 2. So, the these antibodies will recognize MHC class 2 molecules. Now, remember, once you are labeling your proteins, all proteins that are being synthesized will be labeled. So, from this pool of labeled proteins, you need to separate the protein that you are interested in. So, the reagent that is used is a particular antibody. So, you can use specific MHC class 2 antibodies to remove MHC class 2 from the spool of labeled protein. So, that is what is done. So, once you have it, now there are two ways by which you can treat it. One is you do not heat the sample before loading onto the gel. Okay. So, and you just you put it up on a SDS page, on a page and sort of run it, and the other is you heat the samples before you load. Now, when you heat the samples, what will happen is, remember, MHC class 2 molecules consist of alpha and beta chains, and they will fall apart. Okay, and the peptide will be gone because you will not be able to see the peptide in the gel. You will see the MHC class 2 and MHC class 2 alpha and class 2 beta. Now, if you do not heat, then the MHC class 2 molecules will remain together along with the peptide, and so they will run as a complex of MHC class 2 alpha beta, and they are held together. They will migrate with a higher mobility than the ones where you have heated. Okay. So, it is an important experiment to show that that the MHC class 1 molecules, the MHC class 2 molecules will be held together in the absence of heating. And now, if you do these sort of experiments from cells in which there is a mutation that affects MHC class 2, and if the MHC class 2 molecules are unstable, then what will happen, or they are bound to peptides that are not very stable, what will happen is, even in the absence of heating, the MHC class 1 class 2 alpha and the beta peptides will fall apart. And in fact, these are the sorts of experiments that were done to show that there are the molecules that are important for MHC class 2 stability. And in fact, if you can transfect this cell with some of these assembly factors, then you find that yes, you have regained back the ability of MHC class 2 molecules to sort of be associated with peptides and migrate as a complex. Okay, very important experiments, and that is why I initiated this part of the talk with the PulseChase experiments. So, we will discuss MHC class 2 assembly. Now, as MHC class 2 is synthesized, the alpha and beta chains, they come together, they bind and they associate with a particular protein molecule known as invariant chain. Now, why is it called invariant? You see, because for the people in the MHC field, they are so used to polymorphisms and polygenic proteins that MHC class 2 alpha and beta are obviously polymorphic, polygenic, and they associate with an invariant protein molecule, and hence they may name invariant. The other distinguishing aspect about the invariant chain is that it is not encoded in the MHC. It is encoded in a non-MHC location. So, but anyway, it is once it is synthesized, it comes together. What this invariant chain does is that it forms a trimer of trimers. So, so basically it is a nonomer, and you will have 3 MHC class 2 molecules associating with 3 invariant chains and forming this complex. And this what invariant chain is some sort of a chaperone, and it gets the MHC class 1 molecule class 2 molecules together, and then and escorts them to a compartment known as MHC class 2 compartments, or also known as M2C. And M2C is the way usually by which these are described, and these are lysozyme like compartments. Okay. And so, once it goes in there, and their invariant chain is then degraded by cathepsins, okay, and other proteases. So, so the way it is, you have MHC class 2 molecules, they associated with invariant chain, and as I said, they form a trimer of trimers, and then this complex go to the M2C, and it is in the M2C that invariant chain is degraded, except for one part of it, and that is known as the clip. And that is the clip is the class 2 associated invariant chain peptide. So, now you have a complex known as MHC class 2 and associated, that is associated with clip, and that is a sort of a remnant of the invariant chain. Now, how does one get rid of this peptide that is associated with it? So, there is another molecule known as DM. Now, we had come across DM before. Now, DM is encoded in the MHC, and it is the genes are located in the place where the LMPs and the taps are also located. Now, what DM does is it catalyzes the removal of clip, and in fact DM acts as an editor of MHC class 2 binding peptides. So, what DM tries to ensure is that you have very high affinity binding peptides or slow off rate peptides that bind to MHC class 2, and so the MHC class 2 molecules would be stable. And I talked to you about the analogous role of Cathepsin in MHC class 1 that has a similar sort of function. So once DM does the catalysis, so here is a question that I would like to ask you: if in cells, if there is no DM, what would happen? What would happen is that you would have MHC class 2 molecules associated with clip, because there is no DM to take it off. The clip molecule, the clip association with MHC class 2 is not a very stable association. And that is why the MHC class 2 molecules that are formed over here are not very stable. So what DM does is removes the clip off and gets peptides that bind with a slower off rate to MHC class 2 molecules on. Okay. Then once you have now properly well formed MHC class 2 peptide complexes, they sort of egress on and they are expressed on the cell surface. Okay. So, the way MHC class 1 molecules are formed and the way MHC class 2 molecules are formed, they are very different, and you should be a little bit aware even in terms of cell biology, the localizations and all are very distinct, and that is an important aspect and you should think about that. So, what would happen is antigens that are generated within the cytosol, they are by and large the peptides are generated via the MHC class 1 pathway, whereas pathogens that enter via lysosomes or are endocytosed, those peptides are generated in the M2C, and therefore their peptides are expressed by the MHC class 2 molecules. So, what from the host point of view, from the cell point of view, it has taken care of both parts, both the endogenous as the exogenous pathway by which proteins are processed and can enter cells. Okay. So, some differences between invariant chain and DM, which is also known as the editor in chief. So, invariant chain is as the name suggests, it is invariant, and DM there are some polymorphic residues known. Invariant chain is not linked to MHC, this is MHC linked. Where is it? And I think what we will do is I would like to just show this to you. Wait, sorry. Yeah. So, okay. So, this is this is the MHC class 2 region, and these are the proteasome genes, and this is DM. This is where DM, and this part is located between DP and DQ, and over here this part is located between the K and the MHC class 2 locus over here, and you can see this is where DM is, and this is where the proteasome and TAP genes are. So, it is right in the cluster of those 7 genes that were found. Okay. So, the invariant chain, the job of the invariant chain is to take the complex of MHC class 2 molecules and then sort of chaperone them to the M2C, and it does so by certain targeting motifs, and these are Dilucian motifs. Okay. DM on the other hand goes to the M2C by itself and it targets via a tyrosine motif. Now, its job mainly is to remove clip from the MHC class 2 molecules. You see, in invariant chain, once it goes there, its primarily job is done, and therefore and that is why it is degraded. You have to get rid, the messenger is degraded, and a part of the messenger is left on, and that part is removed by DM. The invariant chain, it prevents proteins, peptides to bind MHC class 2 in the ER, and but the main job of invariant chain is to shepherd or chaperone it to the M2C. The main job of the main job of invariant chain is that the main job of DM is that it is the peptide loading capacity of it. So, what would be the phenotype of an invariant chain knockout mice and a DM knockout mice? So, in the case of an invariant chain knockout mice, the MHC class 2 molecules will be stuck in the ER because they would not know how to go to the M2C in the absence of invariant chain. So, there would be no expression of MHC class 2 on the surface. Now, if there is no expression of MHC class 2 on the surface or very low expression, what would happen is there would be no selection for CD4 positive T cells. So, no MHC class 2, no CD4 positive cells, no MHC, no T cells. Okay. But in case on DM, what would happen is MHC class 2 would get expressed, but this would be MHC class 2 that is expressing CLIP, which is an inefficient peptide, but there would be some selection for CD4 positive cells. So, you would have MHC class 2 expression even though it is not the type of MHC class 2 that you would like, but you would have MHC class 2, but the peptides that would bind on would be low affinity peptides, not high affinity peptides. So, the MHC class 2 molecules are not the way that you would want them to, but you would have MHC class 2 on the surface, and you would also have selection of CD4 positives, but it would be an altered selection because the proper right type of peptides are not there on MHC class 2. So, the student should be able to think a little bit about the differences, about the functional roles of this, especially in vivo. Now, to make matters somewhat more complex, you have MHC, you have what is known as DO. Now, you have DM and DO. Okay, both DM as well as DO are MHC class 2 like proteins, okay, but they do not bind peptides, but they have other jobs. DM is, as mentioned, is some sort of an editor in this thing. DO on the other hand, its role is controversial, but what has been shown is that DO and DM are often associated with each other. But the problem with DO is that it is not expressed, its expression level is found only in some type of cells. So, for DM action you really do not DO is not required because you have some cells where there is no DO and still the DM functions and MHC class 2 is expressed. But when DO is there, it modulates DM function. So, and DO is expressed only in B cells and thymic epithelial cells. So, DO modulates DM activity and therefore affects the repertoire of peptides that bind to MHC class 2. Now, in the H2 DO lacking mice, the MHC 2 peptide repertoire is altered compared to wild type, not surprising. What is interesting is that you have an increase in number of CD4 positive single positive cells. So, you have much more single positive cells, perhaps because of alteration in this in peptide repertoire, and maybe selection of CD4 positives is somehow being affected. Now, in recent days what has been found is DO inhibits the entry of activated B cells. Remember, it is expressed in B cells. So, it inhibits the entry of activated B cells to enter germinal centers, because germinal centers you have the T and B cells being activated, and that is where actually the initiation of the immune response takes place. The B cells become responsive to the T cell cytokines, so on. So, DO seems to be affecting that also. Now, MHC class 1 peptides are generated by proteasomes. So, what are the enzymes that that play an important role for MHC, for the generation of MHC class 2 peptides? It turns out there is a family of enzymes belonging to the cathepsins. Now, cathepsins are belonging to the papain family, and over here they are primarily cysteine proteases, which means cysteine is on the active site, except for two of the more famous ones, which is cathepsin D and E are aspartyl proteases, but other than that they are mainly cysteine proteases. What is also known is cathepsin D, E, F, I, S, they are broad specific endo proteases, whereas cathepsin B, H and Z are exopeptidases. Cathepsins are primarily found in lysosome compartments and lysosome or MHC 2 compartments. Now, there for cell biologists there is a difference between lysosomes and the M2C. So, it appears the MHC class 2 are present in specialized molecules which are lysosome like, but not exactly lysosomes, and that is why this name has been given as M2C. Because certain markers that people look for, there are some differences between the markers between the true lysosomes and the M2C compartments. But cathepsins are present in both these compartments, and they are important for the generation of peptides, because once you have proteins over there, they need to be broken down, and cathepsins are primarily responsible for it. Now, in this cathepsin B and D are the most abundant proteases, but they do not play a major role in cleaving invariant chain. Remember, invariant chain shepherds it over there and then needs to be cleaved. Cathepsin S on the other hand plays an important role in invariant chain degradation. It is present in B cells and dendritic cells. It is induced by interferon gamma. IL-10, which is an immunosuppressive cytokine, it suppresses cathepsin S expression. And lack of cathepsin S attenuates several autoimmune disease models in mice. So why would this be? Because there is a correlation over here between MHC class 2 expression and autoimmune diseases. It is something that will be discussed. So what would happen is, if you do not have cathepsin S, it might there might be lesser expression of MHC class 2, which might affect the autoimmune diseases. Now, MHC class 2 peptides can also be generated by cathepsin L, and cathepsin L can in fact it can substitute for some functions of cathepsin S. It is present in macrophages and cortical thymic epithelial cells. Now, here mice that lack cathepsin L have 70% reduced numbers of CD4 positive T cells in the thymus and periphery. They also shed their fur. So clearly over here you can see where cathepsin L seems to be playing a role in MHC class 2, and as a consequence of that it is affecting the selection of CD4 positive T cells. So not surprisingly, cathepsin L lacking mice demonstrate reduced autoimmunity and reduced numbers of CD4 positive cells, because they are sort of linked, and that is something that we will be seeing. Now, apart from cathepsins, other proteases are also involved. Asparagine endopeptidase is shown. It is a cysteine protease that cleaves after asparagine and is important in processing of antigens, especially for example tetanus toxoid, which is a very well studied antigen. So we will just briefly describe the MHC class 2 processing pathway. You have extracellular antigens that come in over here, and so they meet, the peptides are generated in the M2C, and what is shown over here is that the MHC class 2 molecules are synthesized. They bind together with invariant chain, and invariant they form a trimer of trimers, and invariant chain shepherds them to M2C like M2C compartments, where they meet these extracellular antigens or proteins, and then these are cleaved by cathepsins, and part of the peptides are bound to it. Over here you will remember that invariant chain is cleaved, and you have MHC 2 molecules along with clip, and then DM removes the clip and then helps the MHC class 2 molecules load on with higher affinity peptides, and then these are expressed on the cell surface. Okay. Now, we had discussed this aspect about a link between MHC molecules and disease. Now, defects in MHC gene expression lead to immunodeficiencies. So, and the reason for that is MHC molecules are required for both T cell development and activation, and this is something that we have studied in this part of the lecture. And some MHC alleles are associated with susceptibility or resistance to autoimmune diseases. Okay. Now, one important disease is known as a Bayer lymphocyte syndrome. So, and there could be several reasons for the Bayer lymphocyte syndrome. So, what this means is Bayer lymphocytes means there are less number of lymphocytes. Now, what could that be due to? So, for example, if you have defects in TAP expression, so BLS can be due to mutations in several genes. One of them there would be in TAP. So, if you do not have TAP, you do not have MHC or class 1 expression, or even if you have some MHC class 1 expression, those MHC class 1 molecules are unstable. Because you have reduced number of MHC class 1 molecules, you have less selection in thymic selection. So, you have less number of CD8 positive T cells, and these lead to immunodeficiencies. These are respiratory, so you will have respiratory and skin infections. Now, remember over here in this case you are lacking CD8 positive T cells, your CD4s are there. Now, in some cases you have transcription factors which control MHC class 2 expression. So, for example, the very well known one is C2TA. Now, in these sort of animals or humans, this is important for expression of molecules that are important in the MHC class 2 gene expression. So, MHC class 2 molecules, invariant chain and DM, so on. So, if you do not have this, what would happen is there would be no MHC class 2 molecules or very less reduced expression of MHC class 2 molecules. What this would do is you would have less number of CD4 positive cells. In the absence of CD4 positives, which are important in helping macrophages, helping in B cell help, so on, it results in severe combined immunodeficiency. And C2TA is an example of a very important transcription factor that is important in MHC class 2, and since MHC class 2 is so important, it results in severe combined immunodeficiency, and it is a very important bare lymphocyte syndrome. Now, there are some HLA types that are associated with certain autoimmune diseases. So, for example, a particular HLA allele, which is the MHC class 1 molecule, HLA B27, there is a very strong association with spondylitis. So, very strong association with HLA B27 with spondylitis. In fact, the relative risk is fairly high. And you can see some sort of risk along with other autoimmune diseases. So, for example, multiple sclerosis with the DR2 and myasthenia gravis with DR3. There is, but the relative risk here is not as high as the striking one with HLA B27. Okay. So, the other example of a very high risk is insulin dependent diabetes, where where the insulin producing cells are sort of are destroyed by T cells. And again, there is a big association with the DR3 and DR4 MHC class 2 molecules. So, again, as I said, so having those MHC class 2 molecules, what happens is that the chances of a particular autoimmune peptide being presented appears to be high, as a result of which it triggers of autoimmune CD4 positive cells, which kill off certain I mean, in this case there are diabetes producing, the pancreatic beta cells are being targeted and they are being killed off, and as a result of which you have this your risk is high. So, we will try and summarize what we had learnt in these classes. So, in MHC class 1 and class 2, the main important point is that the MHC class 1 pathway is the endogenous pathway, which means proteins that are synthesized in the cytosol, and peptides are generated from these through proteasomes. The peptides are then translocated from the cytosol into the ER by transporters, TAP, and then you have this peptide loading complex where you have the heavy chain and which is bound to beta 2 microglobulin, and then comes and meets them. You have Cathepsin, which helps all this the complex, and you have other proteins like calnex and calreticulin, forms a big complex. And then once you have proper peptide loading on, there is a conformational change, and MHC class 1 molecules are now moved to the surface by vesicular trafficking. The MHC class 2, on the other hand, it samples antigens through the exogenous pathway, and over here the enzymes that are important over here are the cathepsins, and what cathepsins would do is degrade these proteins, the peptides are generated. Now, MHC class 2 molecules come into the M2C with the help of invariant chain. Once they come there, invariant chain is degraded, but you have a part of it attached on, which is the clip, and this clip is removed with the help of DM, and you have DO, which modulates DM activity, but now and DM helps load on peptides with higher affinity, slow off rate onto the MHC class 2. Once this MHC class 2 molecules are loaded with optimal peptides, they sort of move on to the surface. So, one could ask questions: what happens if there is no DM, there is no invariant chain, if there is no TAP, if there is no beta 2 microglobulin, and these are aspects that students should familiar with. Mutations in assembly factors are important, and this is especially true because you have molecules, that these molecules play an important role in diseases. So, for example, bare lymphocyte syndrome, the primary reason for this are TAP deficiencies or C2TA. We also discussed MHC linked diseases. Now, there are certain MHC that if you express certain MHC molecules, there is a high chance of linking with a particular disease. With HLA-B27, you know, there is a very high risk with ankylosing spondylitis. With a particular DRs, there is a very high risk with insulin dependent diabetes, because the pancreatic beta cells are targeted and they are killed off. Now, why is this? What happens is those particular MHC molecules, they express, or they have a tendency to express certain peptides, and those peptides may initiate a CD4 or a CD8 response, autoimmune response. So it's a relative risk is increased if you have some of these MHC molecules. So MHC molecules are certainly very important for initiation, both for T cell selection as well as T cell activation. So it's a very important process. So how they are synthesized, what are the assembly factors, it's very important to understand their pathways. And even now, you know, there are a better understanding of this may help us understand the MHC class 1, class 2 pathways more efficiently. It may help us design better ways to boost up the immune response.